My name is Marissa Schafer, and I direct the Brain and Systemic Aging Lab at Mayo Clinic. I'm an associate professor in the departments of Physiology and Biomedical Engineering, and I have affiliations in the Department of Neuroscience and the Department of Neurology. I direct a Technology Development Award. Our goal is to map senescent cells in the brain using emerging spatial mapping technologies and gold standard technologies.
What drew you to the field of senescence research?
So I did my doctoral training at New York University in Steve Ginsburg's lab, and I was investigating fundamental aging mechanisms in vulnerable brain regions and vulnerable neuronal subtypes that lead to neurodegeneration and Alzheimer's pathogenesis and general brain aging dysfunction. I used calorie restriction as a mechanism to try and suppress those aging processes, and I was really blown away throughout all of that research at how dynamic the network of biological aging processes are in the brain. And I was also really blown away and excited by the notion that these changes are malleable. Through calorie restriction and other interventions, we can suppress aging processes. So this really inspired me to investigate in my postdoctoral training a fundamental aging mechanism that might have impact throughout the body. So I came to Mayo Clinic and I trained in Nathan Milbraster's lab in the Colgat Aging Center. And in that environment, I was able to study senescence and other aging mechanisms across multiple mouse and human tissues. And then I started my lab in the middle of the pandemic with support from a K99R00. And my lab was really built on a foundation that's a really clear merging of my training experiences. So we're studying mechanisms that contribute to age-related cognitive decline with senescence as a central focus in that work.
Can you briefly describe the research that you and your team perform?
We study the cells and cell communications of cell interactions that contribute to vulnerable brain regions that we know are some of the first dominoes to fall in cognitive decline, leading to Alzheimer's disease. So we do this through spatial mapping, which are technologies that enable us to profile the genes and proteins that are altered in specific cells, and that lets us understand or better understand how those cells are communicating or changing their environment. My lab also studies age-related changes throughout the body, so age-related changes associated with disease that can impact the brain through the cells and molecules that are released into circulation that can influence the brain. And whether we're focusing on the mechanisms in the vulnerable brain microenvironments or we're focusing on the mechanisms that are communicated through blood, we're searching for targetable biological processes that we can target through drugs or other interventions to interrupt these dysfunction cascades and ultimately preserve brain and systemic function, so to push back or prevent or slow cognitive decline and other forms of age-related dysfunction.
How does your research impact the field of senescent cell study?
SenNet is tasked with mapping senescent cells across mouse and human tissues. So SenNet is tasked with using both new technologies such as spatial mapping and gold standard technologies and really understanding where are the senescent cells and what are they doing. And my lab is doing just that. So we've been optimizing different new spatial mapping technologies. We've generated a custom gene set that we call the SenBrain signature, which enables us to investigate senescent cells in mice. We're also doing collaborative research to explore senescent cells in the human brain. We share our knowledge and our optimized protocols and our data sets very widely. So we're of course sharing these within the consortium. We're releasing them in preprint form. We're also communicating them through presentations. So one of the goals of SenNet is just to, as quickly as possible, share this information so the greater scientific community can learn from our discoveries as quickly as possible. So we are, without question, doing this.
What would you tell students and junior investigators who are eager to contribute to this research?
So there is just an incredibly vast number of scientists across the globe, not just in SenNet, of course, who are studying senescence and aging mechanisms. And I can't stress enough how important it can be to find good mentors. So I would encourage young investigators to reach out to people within their network or within their state or somewhere where they might be willing to travel to for research experience, either short-term research experience or for a PhD or postdoc, and try to find somebody that you can really click with and build some scientific questions with. So the mentoring relationship is really important. For people who are already in a training mentored environment, I just encourage them to check out the SenNet website. We are constantly releasing our new publications and our preprints. We're constantly releasing new data. Those are really valuable resources that can help one just build their knowledge sets, but also access the data that they can use to develop hypotheses and test questions. I mean, one thing that's so cool about SenNet is we are generating just a huge amount of data that we're releasing as quickly as we can. And that data people can use to mine and ask their own scientific questions. There's so many scientific questions that we're asking, but there's so much more that can be asked by others. So these are freely accessible to the public, which is fantastic.
What are your thoughts on SenNet as a community and resource?
To be perfectly honest, I didn't know how everything would go when I first joined SenNet because I kind of perceived it as, I mean, a lot of science is somewhat competitive, but I was just so blown away by how quickly the SenNet community came together, how collaborative and collegial and open the network has been. It's really astounding, the discoveries that we are making by working together and how quickly we're moving the science forward. So I really think it's a privilege to be part of SenNet, and I'm really grateful to the NIH for supporting this effort.
What inspired you to join the SenNet Steering Committee in a leadership role?
So SenNet is in a really important phase. I mean, all phases of SenNet have been really important for different reasons, but SenNet is in this very important phase of annotating our data, compiling our data, disseminating data, and at the same time, we're publishing our discoveries, we're publishing recommendations for investigators to map or to investigate senescence in their own labs. And I think joining steering committee as a co-chair offered me the opportunity to share my knowledge and my experiences, both in a conceptual way and also in a practical way, and to influence some of the priorities of SenNet, as well as to influence the information structures that we're using to share our data and our information with the public. I will just say to that I mean, that is the priority, is sharing the most useful, the most clear information we can with the public. So anything I can do to help with that, I'm really grateful to do. And it's really a privilege to work with so many exceptional investigators and other individuals to advance that goal.
Please share any further insights into SenNet or senescence research.
I'm very excited about the data, the discoveries that are coming out of SenNet. I'm really excited about our focus on phenotypes. So I think it's really, one of the challenges of senescence is that there are no single biomarkers, or there are no single suite of biomarkers that enable one to detect senescent cells across tissues. So I think we're really tapping into and starting to categorize the different phenotypes, meaning the different features of senescent cells that are the dominant senescent cell fates. So we're really excited to share that with the scientific community. We're also advancing and expanding our analyses to really dig deeper into senescence function, which is something that we've been working on for a long time. We and other groups have been working on for a long time. So I think those are really two really important initiatives and foci of SenNet that are rolling out that I think are going to be really valuable to the scientific community.